Hi everyone, welcome back to my educational channel on biology. In this video, we will discuss the process of pollination and fertilization in plants based on section 6.3 in the Form 5 KSSM biology syllabus. So this video can also be used by those studying other syllabuses where the information may be relevant to them. So in this video, we're going to discuss the process of pollination, which is the transfer of pollen grains from the anther to the stigma of a flower. And next, the pollen tube grows downwards through the style, through the ovary, and then to the ovules. After that is the process of double fertilization that occurs in the embryo sac, whereby one of the male gametes will fuse with the egg cell and another male gamete will fuse with two polar nuclei. So let's get started on this topic. The learning standards are as follows. 6.3 Pollination and Fertilization. Firstly, after this lesson, we should be able to define pollination, which is the process of transfer of pollen grains from anther to stigma. Next, after that, are the other events that follow after pollination. We should be able to describe the formation of the pollen tube and the formation of male gametes. Next, the third process that occurs here is double fertilization. So explain double fertilization in the formation of the diploid zygote and the triploid nucleus. And finally, explain the importance of double fertilization for the survival of flowering plants. So here in this first picture, we see the formation of the pollen tube that grows downwards from the pollen grain into the style. And in this diagram, we see the process of double fertilization that occurs in the embryo sac of the plant, in the ovule of the plant. So let's look at an overview of this chapter, Form 5, Chapter 6, Sexual Reproduction in Flowering Plants. In the first video, we have discussed the structure of the flower, and then in the second video, we discussed the development of pollen grains in the anther and the development of the embryo sac in the ovule. So in this third video, we will discuss pollination and fertilization. That means the event called pollination, followed by the development of the pollen tube on the carpel of a plant, and then double fertilization. And finally, after this video, there will be a last video on the development of seeds and fruits and the importance of seeds for survival. So we are going to discuss pollination and fertilization in this third video. Here's a very good summary of 5 events that occur starting from pollination down to fertilization, okay, until fertilization. So you can copy this summary, which is very useful, into your notebook. Now, after the pollen grains have been released from the anther, okay, that is matured, then it will undergo pollination. That means the transfer of the pollen grains from the anther to the stigma of the same flower or of a different flower. So pollen grains are shown in yellow here. So they can be transferred by wind or water and insects or other animals. Okay. Now, once the pollen grains land on the stigma of a flower, then it will germinate. So germination of pollen grains means the formation of the pollen tube that grows down from the stigma into the stalk. Next, after germination has occurred, mitosis of generative nucleus, number 3. Mitosis of generative nucleus will occur to produce 2 male gametes that are haploid, as shown here. Okay, that the tube nucleus is always in the front, and the 2 male gametes are behind in the pollen tube. Fourthly, there's the growth of the pollen tube from the stigma here at the top to the style and then to the ovule here. Okay, and it's to help the male gametes move through the pollen tube to the ovule. Next, double fertilization occurs in the embryo sac in the ovule here. Okay, so in double fertilization, 2 processes occur. one male gamete, which is haploid, fuses with one egg cell that's haploid to form one diploid zygote, which is diploid. Another male gamete, which is haploid, will fuse with two polar nuclei, which are both haploid, to form a triploid endosperm nucleus, which is triploid. So let's start with pollination, the formation of the pollen tube, and formation of male gametes. Three important events that occur after the formation of the pollen grains. Six point three, pollination and fertilization. First question, describe pollination. So pollination is the process in which pollen grains are transferred from the anther, which is the male reproductive structure of the flower, from the anther to the stigma, which is the female reproductive structure. All right, so because the plant cannot move on its own to search for a mate for the reproduction process, right? So it uses the pollinating agents such as insects, mammals, birds, water, or wind to transfer the pollen grains in the process of pollination. All right, to transfer the pollen grains from the anther to the stigma of the same flower or of different flowers. Okay, it needs a pollinating agent to transfer the pollen grains. Now, next, the presence of pollen grains on the stigma then triggers the process of fertilization, right? So once the pollen grains land on the stigma, then processes occur which will finally lead to the fertilization of the egg cell by the male gamete, right? So after pollination, what actually happens? Let's look at number 4, okay, to explain number 3. How does the presence of pollen grains on the stigma trigger the process of fertilization? So let's look at number 4. The pollen grain will germinate on the stigma after it lands on the stigma. So it will germinate and form a pollen tube, which carries the male gametes. And this pollen tube will grow downwards to the embryo sac in the ovule, inside the ovary, where the egg cell is found, okay? So that the male gamete can enter the embryo sac and fertilize the egg cell, okay? So male gamete also needs a way for it to be transported to the female gamete. So it uses the pollen tube. Pollen tube helps to deliver the male gamete to the egg cell in the embryo sac of the ovule. So now we're going to discuss fully the process of pollination, then germination of pollen, and then formation of male gametes, growth of pollen tube, and double fertilization, right? So take it like a story. So number 1, okay, once upon a time there was an anther, right? So the wall of the anther dries and splits, all right? So the pollen sac in the anther will burst and release the pollen grains, okay? So these pollen grains are then transferred through the process of pollination, right? From the anther to the stigma by pollinating agents such as insects or wind. Okay, so here we see the anther. It releases the pollen grains when the pollen sac bursts, and then these pollen grains are transferred either by insects, wind, or water from the anther to the stigma of the same flower or of a different flower, right? So the pollinating agents can be insects, wind, mammals, water, and so forth. Now let's go on to number 3. We will try to answer this question. Describe the formation of the pollen tube. So once the pollen grain has landed on the stigma, the next process is the formation of the pollen tube. How to describe it? So germination of pollen grain on the stigma is stimulated by a sucrose solution secreted by the stigma, which will cause the pollen grain to grow a pollen tube on the stigma. So the sucrose solution provides the food and in that way provides the energy for the growth of the pollen tube, right? So this pollen tube will then grow down from the stigma to the style and then down to the ovary, and finally it will reach the ovule so that the male gametes can carry out double fertilization, right? Now third question is describe formation of male gametes. So after the pollen tube starts to grow downwards, a process occurs, which is mitosis of the generative nucleus. So the generative nucleus will divide by mitosis to form two male gametes, which are haploid, right? So at first there's only one generative nucleus, but after that, after mitosis, there'll be two male gametes. All right. Now the tube nucleus is always further down near the tip of the pollen tube, and it guides the pollen tube down to the ovule and to the embryo sac. So the generative nucleus undergoes mitosis to form two male gametes. Now next, what happens? The pollen tube will grow downwards to the ovule. Okay, it will grow through the style and through the ovary here to reach the ovule. Now, as it grows, the end of the pollen tube or the pollen tube end will secrete enzymes to digest the tissue in the style so that it can grow downwards. And it will allow the male gametes to also move downwards together with the tube nucleus. So tube nucleus will guide the pollen tube to the ovule, and the male gametes will follow behind. Next, the pollen tube reaches the embryo sac in the ovule. It will penetrate the ovule through the micropyle or small opening here. Then the tube nucleus degenerates. So now you cannot see any more tube nucleus because it has degenerated or broken down, leaving the two male gametes, right? And the pollen tube bursts to allow the male gametes to enter the embryo sac here, which contains the eight nuclei, right? So next, a very important process occurs, which is called double fertilization, because it involves two male gametes. That's why it's called double fertilization. Now, the first male gamete fertilizes the egg cell to form a diploid zygote. The blue arrow here, okay? Whereas the second male gamete fuses with two polar nuclei in the center part of the cell to form a triploid endosperm nucleus, 3N. Okay, because each nucleus is haploid, so three nuclei will become a triploid condition. Okay, so the final diagram shows you the two male gametes fusing with the, for example, the first male gamete will fuse with the egg cell, and the second male gamete will fuse with the polar nuclei. So let's look at this learning standard. Explain double fertilization in the formation of the diploid, zygote, and triploid nucleus. Okay, so this first diagram here is from the previous slide. Shows the male gametes fusing with the nuclei in the embryo sac. All right. So what is the end product? Now, the first male gamete fuses with the egg cell here, the pink cell, and it will form the zygote, which is a diploid cell. All right, that will in the end become the embryo. Now, the second fertilization process involves one male gamete and two polar nuclei. This will produce a single cell called the triploid endosperm nucleus, right? And this will divide by mitosis to form many cells that store food. So that is called the endosperm or food store. Okay, let's read from here. Now, let's repeat what we have discussed just now. Double fertilization occurs whereby one male gamete here fertilizes the egg cell to form a diploid zygote. The second male gamete fuses with two polar nuclei to form a triploid endosperm nucleus, right? Then after double fertilization, the diploid zygote develops to form the embryo or baby plant, whereas the triploid endosperm nucleus divides by mitosis to form the endosperm tissue here, right, which is the food stock. We'll be discussing more of this structure in the next video. So here is a popular question on double fertilization. Describe the events that occur from pollination until double fertilization in angiosperms. 10 marks, okay, for essay question. And here's the answer. So we have discussed this in the previous slides, and you can copy down this answer for this question in a notebook. So let's go through it, and I've blanked out certain parts. So you can try to answer this question first and fill in the blanks, all right, before we discuss the answer. You can pause the video for a while, okay, and write down the answer for each part. So let's look at this answer here. What are the events that occur starting from pollination until double fertilization in the flowering plant? Firstly, the wall of the anther containing the mature pollen will dry and split. So what is released from the pollen sac? Yes, pollen grains. Now, what is the term for the transfer of pollen grains? It is pollination, right? And the pollen grains are transferred from anther to stigma by pollinating agents such as wind, insects, and water. Fourthly, the pollen grains germinate. Okay, so this is a keyword, germinate on the stigma. And to germinate means it forms a what? Pollen tube, which grows down through the style towards the structure here called the ovule. Germination of pollen is stimulated by secretion of what from stigma? Yes, sucrose. Okay. Now number 5, the generative nucleus moves along the pollen tube towards the ovule, and it is led by the tube nucleus in the front. It will then divide by what? To form two haploid what? Divides by mitosis. To form two haploid male gametes. Now next, the pollen tube secretes something to digest the tissues of the style. What is that? Enzymes. Number 7, the pollen tube reaches the what in the ovule? This structure here, pink cell here, embryo sac. It penetrates the ovule, okay, penetrates the ovule here through a small opening here called the what? Micropyle. Then the tube nucleus. What happens to the tube nucleus that was leading the way? It degenerates. The two male gametes then enter the embryo sac. The two male gametes here will enter the embryo sac. Now, what process occurs next? Number 8. Double fertilization. One male gamete fertilizes the what to produce a diploid what? One male gamete fertilizes the egg cell to produce a diploid zygote. The second male gamete fuses with what? To form what? Second male gamete fuses with two polar nuclei to form a triploid endosperm nucleus. Okay? So this is the final answer for the essay question. What happens from pollination until double fertilization? So make sure you memorize these processes, and you can divide them into 5 parts. First is pollination. Secondly is germination of pollen grains on the stigma to form a pollen tube. And then thirdly is mitosis of generative nucleus. Fourthly is the pollen tube will grow downwards to the ovule and penetrate it. And fifthly, double fertilization occurs. Okay, 5 different events. Okay, so in this section B, we are going to repeat what we have mentioned earlier in the previous slides. Okay, the process of double fertilization and the formation of the diploid zygote and the triploid nucleus. We already discussed it in the previous slides. So summary on double fertilization. So it's good to repeat again and again what double fertilization is, okay, in order to understand and remember it. Double fertilization. What is double fertilization? Explain this process. So double fertilization involves two male gametes in which the first male gamete fertilizes the egg cell to form a diploid zygote. This is 1 mark. And secondly, the second male gamete fuses with two polar nuclei to form a triploid endosperm nucleus. This is another mark. Okay, to explain what is double fertilization. Now, so as a summary, you can even write this process of double fertilization as an equation to help you memorize. Okay, you can copy this down, these two equations down, and then stick it on the wall in your bedroom and try to memorize, review it from time to time to memorize it. So firstly, one male gamete, which is haploid, fuses with one egg cell, which is haploid, to form a diploid zygote. This is one process of fertilization. Secondly, one male gamete, the second male gamete, which is haploid, fuses with two polar nuclei, which are both haploid, to form one triploid endosperm nucleus, which is triploid. So this is the other process of fertilization. So that's why it's called double fertilization, because two processes of fertilization occur, okay, in this process. And here's a picture which shows you double fertilization. The first male gamete fuses with the egg cell to form the diploid zygote, and the second male gamete fuses with two polar nuclei to form the triploid endosperm nucleus. Sometimes it's just called triploid nucleus, okay? But we add the word endosperm because in the end, the triploid endosperm nucleus will divide by mitosis to form many cells that in the end form the endosperm or food stock, all right? So you must memorize how to explain double fertilization. This is the most important question in this lesson. Okay, let's come to the next uh, title and the next question. Explain the importance of double fertilization for the survival of flowering plants. So double fertilization is known to be a very important factor in the survival of flowering plants, right? So how does double fertilization help the flowering plants to survive, to live on, and for the plant species to continue to exist on Earth? Another important question. Discuss the importance of double fertilization for the survival of flowering plants. 6 marks. It can be an essay question. What is the importance of double fertilization to the flowering plants? How does it help them to survive? So there are two parts to this. It's talking about the fusion of the male gametes with the different uh, parts of the embryo sac. Okay, so the first one is the fusion of one male gamete with the egg cell to produce a zygote, which then later on undergoes mitosis to become the embryo or young plant. So what is the significance or importance of this process? Now, it helps genetic information to be passed down from the parent or from the generation of the parents, right, to the next, which is the offspring. It helps genetic information to be passed down from parent to offspring. Okay, how does that happen? Okay, the male gamete contains its own set of chromosomes, which comes from the male plant, and the female gamete or egg cell also contains another set of chromosomes, which comes from the female parent, right? So during fertilization process, the two sets of chromosomes from the two gametes will combine together in the zygote, right? So in that way, the genetic information from both parents, which is carried by the chromosomes, right, is passed down from the parents to the offspring. Okay, so the male parent will contribute one set of chromosomes that contains the genes to determine the characteristics of the organism. The female parent also passes down one set of chromosomes in the egg cell, okay, to the offspring to determine the characteristics of the offspring. Okay, so the zygote contains two sets of chromosomes that contain genetic information. One set of chromosomes is from the male parent and one set from the female parent. Okay, so that's how genetic information is passed down from one generation to the next. Next, the fusion of the male gamete, the egg cell, to form the diploid zygote will restore the diploid condition with the formation of a diploid zygote from haploid gametes. Okay, so the haploid gametes are formed through meiosis, and they have only one set of chromosomes each. So when these two gametes fuse together, when two haploid gametes fuse together, then the diploid condition is restored. Okay, is brought about in the diploid zygote. So this diploid condition is very important to be maintained, as the parent plant also is a diploid organism, right? So the zygote should also be diploid. So most of the cells in the parent plants are diploid, except for the gametes. Now, secondly, the fusion of the second male gamete with two polar nuclei produces a triploid endosperm nucleus. So what is the significance of this process? So this triploid nucleus will then undergo mitosis to form the endosperm tissue which surrounds the embryo. So this endosperm tissue is not the offspring. It is just a food stock that contains starch, oils, and proteins that can be used for the development of the embryo, especially during germination, all right, or even if there's no germination, for the survival of the plant species. So if this embryo doesn't have the endosperm tissue or food store, it may not be able to germinate or live long. Now, next we come to two types of flowering plants, the eudicots and the monocots. So in eudicots, such as legumes, the bean plants like the green bean, mangoes and mustard, the endosperm is fully used up by the embryo to develop before the seed matures, right? So the endosperm is fully used up. So when the seed has matured and is formed completely, then the mature seed will have no endosperm because all the endosperm has been used up by the embryo for development. So what stores the food is the two cotyledons that are produced through mitosis of the zygote. Okay, so two cotyledons, they will get the food from the endosperm and they will store that food. So they will serve as the food storage tissue for eudicots. So eudicots usually do not contain the endosperm. They contain the two cotyledons, which are usually large in size, and these cotyledons store the food for the germination of the seed and for the survival of the seed. So here we see a bean plant, which is an eudicot. And here you see the scar, which is the hilum, called the hilum, where the punicle or the stalk was attached to the ovule previously. This is the micropyle below here that allows air or oxygen and also water to enter the seed so that the embryo can germinate. And next, here we see the longitudinal section of this bean plant. You can see two big cotyledons here. So the embryo consists of the plumule, it's a young shoot, the radicle, the young root, and then two cotyledons here. Okay, and outside here is the testa or seed coat. Then here is the micropyle. So the second type of flowering plant is called the monocots. Mono means one, one cotyledon per seed. Okay, whereas eudicots, the D there means two cotyledons per seed. So in eudicots such as coconut, wheat, barley, corn, only a part of endosperm is utilized for the development of an embryo. So some of the endosperm is still present upon germination. That means only a part of the endosperm is used for development of the embryo. So when the seed matures, there's still some endosperm left. Okay, until germination, when the endosperm will be used for the seedling to grow. So upon germination of the seed, the thin membranous cotyledons would absorb the stored food from the endosperm so that the seed can grow. The young plumule or young shoot can grow and the young root or radicle can grow. Okay. Now, if there's no germination, the endosperm tissue will be also used to enable the embryo to survive in the seed for a long time because the embryo will still need to carry out respiration. So you need food in order to carry out the respiration, to break down the food and get the energy to survive, right? So it can survive for a long time because of the endosperm tissue that provides it the food. If conditions are not favorable for germination to occur, right? So the diagram here shows the corn seed, which is a monocot, corn or maize or jagung. So sometimes you can eat the jagung in a cup, right? You can buy from the stall in the mall, and you can see that the jagung has quite a juicy part here, which is yellow in color, that's the endosperm. So we're mostly eating the endosperm of the maize, right? So endosperm takes up a lot of space, a volume in the corn seed. And then here we have the one cotyledon, the white part, and the embryo is at the side here with its plumule or young shoot and the radicle or young root. Okay, so the endosperm tissue is a triploid tissue, but the cotyledon is a diploid tissue that uh, is produced by mitosis of the zygote. Then outside is a tough seed coat. So this diagram shows the bean seed and the corn seed side by side. It's just like what we discussed just now. And you can see here the bean seed, which is an eudicot, okay, has two cotyledons but no endosperm. The corn seed, which is a monocot, has one cotyledon here, the white part, and also has a large portion of it is the endosperm. Okay. Then of course both seeds have the embryo, right, which is made up of the plumule and the radicle, right? This is the embryo here. So here we see the germination of an eudicot seed, such as the bean plant. Okay, so this is some extra information to understand the function of cotyledons. Here we see the seed start to grow the young roots, and it's also in the process of growing the young shoots. Okay, so as it grows, it will use the food stored in the cotyledons here. Now let's look at the function. Function of cotyledons in eudicots, such as the bean plant. Food stored in the cotyledons is used to produce energy through cellular respiration when the food is broken down. All right, so the energy is released for the growth of the seedling, which involves cell division by mitosis. So this process needs energy until the first green leaves are formed. Okay, until after some days, maybe the first green leaves are formed here, right? So the cotyledon would have done its part to, or carry out its function to provide energy for the growth of the plant before the first green leaves are formed. Now in monocots such as corn, it is the endosperm, not the cotyledons, that act as the food store to provide energy through cellular respiration. Okay, now here we see the first green leaves formed already, which can carry out photosynthesis to produce organic food substances like sugars for the growth of the plant. So the function of the cotyledon has already been carried out. So next it will shrink or shrivel, and then it will drop off from the seedling. Okay. Some interesting facts, extra information on the endosperm. As we know, the endosperm is a triploid tissue produced in seeds of flowering plants after the double fertilization process in the embryo sac of the ovule. Now it is triploid or 3N, meaning it has three chromosome sets or three sets of chromosomes per nucleus. So what's the function of the endosperm? It is a food store that provides nutrients such as starch, proteins, and oils to the growing embryo, and it also supplies energy for seed germination. Okay, after the embryo starts to grow and then produces the young roots and the young shoots. Okay, until the first green leaves are formed. Okay, the seedling will depend on the food store such as the endosperm before the first green leaves are formed, which can carry out photosynthesis. So it's needed for seedling development, okay, before the first green leaves are formed. Now, as for its importance to humans, the endosperm is a source of nutrients and food to humans. The endosperm is the most important plant product for humans, and 2/3 of all human calories come from the endosperm. So we depend on the endosperm to provide us with energy through cellular respiration. So that's why we grow cereal crops such as wheat, rice, barley, and corn, is to get the edible seeds or grains, which is mainly endosperm tissue. All right. So this endosperm or food store in the seed provides mainly starch, which is a source of energy. Example, white rice consists of mainly the endosperm without the bran or the germ. And wheat endosperm is ground into wheat flour to make bread. Right? So white rice, let's have a look at a grain of white rice. What does it contain? Now, the outermost shell or layer is called the rice hull. It protects the grain and it's quite tough. Then the second layer is the rice bran, which is fibrous. So to form white rice, the rice bran will be polished off or milled off. But if it's left on, then it will form the brown rice, which is brownish in color because of the bran. So the bran also contains fibers and nutrients. Then the white part here is the endosperm. So the white rice is actually made up of mostly the endosperm, which forms 75% of the seed. And the endosperm is starchy, okay, it's sometimes called the white rice kernel. So it's the food stored in the seed. It helps provide energy to the seed or to the seedling when it's developing, and also provides energy to us, right? When we cook the white rice and eats it. So the germ is the embryo that will form the young plant. Okay. So in the white rice, the germ may be polished off or milled off. Now, lastly, we're going to understand some scientific terms. What are eudicots and monocots? What are the differences between eudicots and monocots? And then what do we mean by haploid, diploid, and triploid condition? So what are the eudicots and the monocots? These are two main groups of angiosperms or flowering plants. So let's compare the eudicots and the monocots. Eudicots, such as the bean plants, have seeds that have two cotyledons per seed. Okay, there are two cotyledons per seed and usually no endosperm. Monocots, such as the corn plant, in each of its seed there is a one cotyledon only, that's why it's called monocot, and it usually has some endosperm left, right? Then the root. Eudicots have a taproot, which is a main root that grows downwards, and then with the side roots branching from the taproot. They don't have a taproot, okay? They have many fibrous roots, small fibrous roots that grow out from the plant, grow downwards from the plant. Then looking at the leaves, this is a very simple way to differentiate eudicots and monocots. Look at the pattern of the veins. If the veins show a network pattern, okay, net-like pattern, so if it shows a network of veins in the leaves, then it's an eudicot. Whereas for monocots, the veins are arranged in a parallel pattern, all right, almost parallel to each other, and the leaves are usually more elongated. Now, in the stem, if we get a cross section of the stem, we will see that the vascular bundles consisting of xylem and phloem are arranged in the form of a ring, circular form. But for monocots, in the stem, the vascular bundles are scattered, right, and not arranged in the form of a ring. Next, let's discuss the condition of the cell. That means the number of chromosomes in the cell. So let's differentiate between the haploid cell, diploid cell, and triploid cell. A haploid cell or a haploid nucleus has only one set of chromosomes. One N, N means one set, N means a set. One N or N. For example, a plant species may have 10 chromosomes in the haploid cell. Examples of haploid cells are the gametes, namely the male gametes and the female gamete, which is the egg cell. So each male gamete is haploid, each female gamete is haploid. All right, meaning that they have in each gamete there's only one set of chromosomes of different lengths. Okay, the chromosomes are of different lengths. They are different types of chromosomes and they have different genes in them. All right, so these are the examples of this is an example of one set of chromosomes. Different chromosomes with different genes to determine different characteristics. So examples are the male gametes found in the pollen tube here, right? And below is the tube nucleus, which is also haploid. And then we have the egg cell here inside the embryo sac here. So this is also haploid. Now, the haploid cell is very important because when two haploid cells or two haploid gametes fuse together, then they will restore the diploid condition in the zygote. Okay? Because n plus n equals to 2n. Now, let's go on to the diploid cell or nucleus. It has two sets of chromosomes, or 2n, two sets, so 2n. For example, if the haploid cell has 10 chromosomes, the diploid cell will have 20 chromosomes because there are two sets of chromosomes in them, two sets of 10 chromosomes in them. Examples of cells or even organisms that are diploid are the parent plants. Okay, even humans also consist of diploid cells, okay, diploid somatic cells. And then zygote is formed from the fertilization of the female gamete by the male gamete, so it's also diploid. And then when the zygote undergoes mitosis, it forms the embryo, which is also diploid, right? And the cotyledon in the plant actually is produced from the zygote that undergoes mitosis, so it's also diploid. Cotyledon is a food stock, right? So all these are diploid cells. Then we come to the term triploid cell. It has three sets of chromosomes, 3n, in the cell, in each cell. For example, if the haploid cell has 10 chromosomes, then the triploid cell will have 3 times 10, 30 chromosomes, 3 sets of chromosomes. All right, now how to get 3 sets of chromosomes? The triploid cell is produced when the two polar nuclei here fuses with one male gamete. Okay, so the male gamete will contribute 1 set of chromosomes, then the two polar nuclei will contribute 2 sets of chromosomes altogether. Okay, so altogether the total is 3 sets of chromosomes, okay, to form the triploid cell. Then the triploid cell undergoes mitosis to form a lot of cells which are also triploid. So this will form the endosperm tissue which stores the food. All right. So here we see the formation of the triploid nucleus when the male gamete fuses with the two polar nuclei, okay, to form the triploid endosperm nucleus. And then this male gamete fuses with the egg cell, so n plus n equals to 2n. So the zygote form is diploid. Okay, and then the conceit we can see the endosperm tissue is actually a triploid tissue, and the cotyledon tissue that is formed from the zygote is actually diploid. So there's a difference between this endosperm and cotyledon tissue. Now, formative exercise is the last part. So let's look at some questions in this formative exercise that are similar to those in your textbook. Now let's look at question 1. Define pollination. 1 mark. So pollination is the transfer of pollen grains from the anther to the stigma, right? Transfer of pollen grains from anther to stigma here. Now, pollination is very important to ensure that reproduction in flowering plants occurs successfully. Explain. So, important to ensure reproduction. So, what is the importance of pollination where reproduction is concerned? Okay, so pollination helps to transfer the pollen grains from the anther to the stigma. And after that, the presence of pollen grains on the stigma will finally lead to the double fertilization process. So that's why it's important. Then to elaborate further, you can say that the pollen grains, when they land on the stigma, they will germinate and grow a pollen tube, which allows the two male gametes in it to move okay, here, to the embryo sac in the ovule here to fertilize the egg cell, so that double fertilization occurs, right? One mark for each statement here. Next question. During formation of the pollen grain, the microspore mother cell divides by meiosis and produces a tetrad. State the feature or the characteristic of the tetrad, the special characteristic, and the importance of such a feature. 3 marks. So here's a picture of the process of meiosis in the formation of the pollen grains. Okay, one microspore mother cell, which is diploid, in the anther undergoes meiosis to form a tetrad, meaning a group of 4 haploid microspore cells, which will after that separate from each other, right? So the tetrad of 4 microspore cells. Now a tetrad contains 4 microspore cells, which are haploid each, having 1 set of chromosomes per cell. It's to maintain the diploid number of chromosomes in the zygote after fertilization or after fusion between the haploid male gamete and the egg cell, right? So the importance of the tetrad is that it contains microspore cells which are haploid, having 1 set of chromosomes, so that after fertilization, double fertilization has occurred in the embryo sac in the ovule, then the 2 haploid gametes, the male and female gametes, they will fuse together and they will form back they will have produce the zygote, which has the diploid number of chromosomes in the zygote, which has the diploid number of chromosomes just like in the parent plant, in the cells of the parent plants. Okay, so in this way, the haploid cells help to maintain the diploid number of chromosomes in the zygote after fusion between the 2 haploid gametes. So let's continue our formative exercise. Now you can pause the video and try these 3 questions first. All right. So it's important to know what stage that they are talking about in the reproduction of the plants. You must determine that before you answer the questions. Right, let's look at the answers. So the first question, the generative nucleus divides by mitosis in the pollen tube. State the importance of this process. So to recall back, the generative nucleus divides by mitosis when pollination has occurred. After pollination has occurred and the pollen grain is on the stigma, right? It grows a pollen tube, and then during this time, the generative nucleus will divide by mitosis. So what is the importance of this process? So to know the importance, we must know the final product of this process of mitosis. Now, it produces 2 haploid male gametes, okay, through the mitosis of the generative nucleus. So this produces 2 haploid male gametes, which are then involved in the double fertilization process. So this is the importance of the process. Now, if more marks are given, you may have to elaborate and explain the function of the male gametes. For example, you can mention the first male gamete fuses with the egg cell to form a diploid zygote, and the second male gamete fuses with 2 polar nuclei, the 2 polar nuclei to form the triploid endosperm nucleus, okay, which is then will form the food stalk. Now, number 5, the megaspore cell contains 8 nuclei. How does this occur? So recall back, where is the megaspore cell found? Now, the megaspore cell is actually the embryo sac, okay, so it's found in the ovule of the plant. And how does it contain 8 nuclei? So a process occurs, right, which is mitosis. The nucleus of the megaspore cell or embryo sac undergoes mitosis 3 times to produce the 8 nuclei. So from one nucleus, after the first process of mitosis, it will divide to form 2 nuclei. And then in the second process of mitosis, the 2 nuclei will divide to form 4 nuclei. And after the third process of mitosis, the 4 nuclei has divided to form 8 nuclei in the final embryo sac that is mature. Now, number 6, state the role of double fertilization in ensuring the survival of flowering plants. The role means the importance of double fertilization. How does it ensure the survival of flowering plants? So we must think back about the products of double fertilization, right? So first fact, the fertilization of the male and female gametes, or the male gamete with the egg cell, produces a diploid zygote. So what is the importance of that? This transmits genetic information to the next generation. All right, that means the male gamete has genetic information from the male parent carried by the chromosomes. Female gametes also have chromosomes which contain genetic material or information from the female parent. So when the male and female gametes fuse together to form the zygote, then the information from both parents is transmitted to the zygote, which is the next generation, which will form the young plant. Okay. Now, next, the fusion of the second male gamete with the two polar nuclei produces a triploid endosperm nucleus that will develop and undergo mitosis to form the endosperm tissue. What is the importance of the endosperm tissue? Explanation. This tissue supplies food for seed germination. Okay, so this now question 6 is quite important to explain the role of double fertilization in ensuring the survival of flowering plants. 1 mark for each point here. Right, that's all for this lesson. Please share, like, and subscribe to my channel and also comment. Thanks for viewing and goodbye for now.